Before passaging the cells, it is important to check its confluency and its morphology for the presence of spontaneous differentiation. The ideal confluency to passage iPSCs is of 70%. Areas of spontaneous differentiation should be removed before starting passaging the cells. iPSC colonies have a compact center compared to the edges. As colonies grow, some might fuse together, giving rise to a phase-bright contrast in the center of the colonies, as cells start to pile out. To remove dead cells floating in the dish, remove media by vacuum aspiration and wash the cells once with warm DMEM. Next, add 2 ml of gentle cell dissociation reagent and incubate for 5 minutes at room temperature. While the cells are incubating with the reagent, label the new Matrigel-coated dish with the appropriate information, such as the cell line name, your name, the date, the media used, and any other information you deem important. Check the cells under the microscope. As dissociation reagent acts, cells will start separating from each other and will appear more loosely attached, especially on the edges of the colonies, where we see that there is a light coming through. Once you observe this morphological change, remove the dissociation reagent by vacuum aspiration. Aspirate the reagent from a corner of the dish to avoid scratching the cells. Wash the cells with 2 ml of DMEM F12 with anti-anti, and then remove media by vacuum aspiration. Next, add 2 ml of DMEM F12 with anti-anti with a 5 ml serological pipette and start scraping the cells using a sterile cell scraper. To detach as many cells as possible, make sure to scrape all of the dish surface containing cells. Transfer the cell suspension to a 15 ml conical tube using a 5 ml glass serological pipette. Then repeat the previous step once more in order to collect all the cells. This time, when collecting the cell suspension, tilt the dish and with the glass serological pipette, wash the cells down and transfer them to the 15 ml conical tube. Pipette up and down a few times to mix. Now that we collected all the cells, we transfer a fraction of the cell suspension to another tube. This will be used for passaging. The volume of cells we take at this step depends on how many dishes we want to plate, the size of these dishes, and how confluent the cells were before passaging. Centrifuge the tubes for 3 minutes at 1,200 RPM to pellet the cells. After centrifuging, a cell pellet will be visible at the bottom of the tube. Before resuspending the cells, gently aspirate supernatant without disturbing the pellet. Using a 5 ml glass serological pipette, add 3 ml of iPSC media and gently resuspend the cells until small cell clumps are still left. Pipetting slowly 3 to 5 times using a glass serological pipette is usually enough. Using a 5 ml glass serological pipette, transfer the cells to the Matrigel-coated dish. Remember to aspirate the Matrigel coating solution before adding the cells. Then, swirl and rock the dish to evenly distribute the cells. Add ROCK inhibitor to the cell suspension at a 10 micromolar concentration. Under the microscope, verify cell clumps density. If the clumps are still too large, pipette cell suspension up and down a few times using a serological glass pipette to break them down further. If cell density is too high, you can remove some of the cell suspension from the dish and add fresh medium to complete its volume. On the contrary, if there are too few cells, add more cells in case you have leftover cell suspension.